Okay, so I think we're going to get started. Good morning, everybody. Thank you for being here. This is the lung map update meeting. I'm SWOG. I'm a thoracic oncologist from Fox Chase Cancer Center and the current PI of the program. My co-PI, Dr. Karen Rickham, is going to be virtual. She could not be here today because of a conflicting meeting. I'm also joined by Dr. Mary Redman, biostatistics head for lung map, among other things. Before we start, besides thanking all of you for being here and everybody online, I'd like to recognize leaders and senior members and Professor Emeritus of this program, Dr. Roy Erbs. Roy, we can just wave. We're recognizing your activity, Dr. David Gandera. Both of them have been instrumental in getting lung map up and running. So thank you for being here. So we have a packed agenda. We have a number of our colleagues online also. So some of the presentations are going to be virtual and some are going to be in person. We're hoping to have adequate time for question and answers at the end of the meeting. And for those of you who are virtual, I think there is a function for you to submit your questions on chat. I don't promise to be able to get to everybody's questions during the meeting, but we'll do our best to answer everyone's questions. So lung map is almost 12 years old. I think for those of you who've been involved with the program, you recognize the name, you recognize what this group is all about. Our goal hasn't changed. We still want to be able to develop novel therapeutic options for patients with advanced non-small cell lung cancer, regardless of histology, second-line space. We are a public-private partnership, and I think that is something to be emphasized. We have enjoyed significant collaboration and close work with a number of our partners in the industry side, many of the academic centers, and obviously our friends at the FNIH, the FDA, and the NCI. So this has really been the village that we need to advance treatment options for lung cancer. The challenges that we're facing is very familiar to those of you who are running clinical trials offices. Budgets are tight, personnel are hard to find. And those are some of the issues that we've heard over the past 6 or 7 months from many of our sites, and we've experienced it at our own centers. Some of these are beyond our control, unfortunately, personnel and issues like that. Some we can control. We have heard loud and clear from everyone involved in trials that they want simpler studies. They want studies where a patient can go on without the need for a lot of testing and a lot of CAT scans and a lot of this and that, and we hear you. We would like to have studies like that, and since we are in charge of writing these protocols, that is what we are going to concentrate on. We fully understand that if you are having staffing issues, the less time you have to devote to screen someone and get them on a study, the more time you have to actually enroll patients on clinical trials. So that is going to be everybody's goals from this point on, to make these studies as simple as possible. I cannot promise that we can make every protocol look like Pragmatica. I think that's a very unique situation. But we would like to have a 3-page consent and an 11-page protocol when feasible and possible. But again, we have a network of partners that we work with, and we have to consider everybody's needs. Patient needs come first, and again, we are going to concentrate on that. But your collaboration and cooperation, giving us feedback when a protocol is about to be released, and you all have an opportunity to look at it, telling us what works for you and what doesn't. All of these are important. You know, we're only an email away, seriously. We might not answer you immediately, but if you send us something that says this is not working for us, we'll work on it and we'll come back with a solution, because we want you to be part of this program and we want more people to join us. So don't be shy about telling us, you know, what we're not doing right, at least from your perspective and from your site's perspective. Again, I won't be able to find you staffing, and I'm not going to be able to find a better budget for your clinical trials office, but there are other things that we can do to make life a little bit easier. So please get in touch with us. Let us know what you need, and we will do our best. LungMap has been one of the most successful platforms for inclusion of patients who we normally have not been seeing on clinical trials by virtue of being available in rural areas, in urban areas. There's a recent publication in JCO Precision Oncology. I encourage you to read it. There is a very nice editorial sort of highlighting the fact that we are accessing patients who, again, normally would not have been involved in a lot of the clinical trials that we run in the world of lung cancer. So there are a lot of benefits for being a member of this platform and this group. So I don't want to take all the time talking about LungMap again right now, but I do like to go through is what we're doing now, what are the sub-studies that we have, and again, open it up to questions and comments towards the end of the program. With that in mind, I'm going to ask Dr. Redmond to provide us an update on the manuscripts. Yeah. So if you want to use the podium, that'd be great.
Okay. Good morning. Let's see, here we go. Okay, so there's our agenda, which we know a little bit about. Welcome, accomplishments, publication updates. Okay, so we have had a very productive 2023. I want to highlight some of the publications that we have out and some things that we have in the works that will hopefully come out in the near future. The first manuscript I'd like to highlight is for those of you who've been around for a while, LungMap S1400I was one of the original sub-studies of LungMap, which was a randomized phase 3 evaluating nivolumab combined with ipilimumab versus nivolumab alone for squamous cell patients who were immunotherapy naive. And part of that project included patient-reported outcomes and quality of life assessment. So the first paper highlighting here by Dr. Unger and all, we published those results. And the nice thing is just that there wasn't any major differences in quality of life, which given that nivolumab and ipilimumab is a combination immunotherapy, that was a significant result. The next paper I'd like to highlight is a paper by Dr. Xing Li at Columbia. And what she did was develop a novel approach for visualization of adverse event data in immuno-oncology. And she also used the 1400I study. And so that is a really nice paper showing how you can highlight immune-related and other adverse events in a way that's a little bit more intuitive. But then the third paper, which I'll talk a little bit more after this part, is the paper that was being highlighted by Dr. Borgai just now, which was led by Dr. Riha Vaidya. She's a health economist at Fred Hutchinson Cancer Center and part of the SWOG Statistical Center with me. And she and to talk about the genesis for this paper that looked at the representativeness of patients enrolled onto LungMap. When we first developed LungMap, about a decade ago, one of the major ideas was that we wanted to be able to bring these investigational treatments to patients all around the community. And a major goal was that the patients that we were accessing were more representative of patients with the disease. And so this work was to basically see, did we do what we wanted to do, which was reach patients with lung cancer in a more representative way. And I'll highlight it a little bit more, but the shortcut to the answer is, in fact, yes, we have been successful. And why the NC National Clinical Trials Network and studies like LungMap are important are because we are better at doing this because we have an established infrastructure. And then another exciting set of work that I'd like to highlight, also looking at the 1400I study, is that partnering with the CMAX, we did some work looking at features that are associated with immunotherapy benefit. And for those of you who attended the translational plenary yesterday, you saw this work highlighted that, as Dr. Vastuba discussed, that it's not just where the immune cells are in the tumor, but it's the location, and that's related to treatment efficacy for patients receiving immunotherapy. So that paper is in press and hopefully will come out soon. So we had a lot of work. None of it was a clinical trial primary publication, but all the ancillary work that we try to do within the lung map infrastructure. We have a lot of papers that are in progress, and I think we expect that most of these will be published within the next year. The S1900A study, which evaluated rucaparib in patients with LOH positive or BRCA positive non-small cell lung cancer. It was presented by Dr. Reese at ASCO in 2021, and the paper is in development. S1900C, which evaluated talazoparib, a PARP inhibitor, combined with avelumab in patients with STK11 genomic alterations in their tumors. That manuscript was presented, the paper was presented by Dr. Skoulides at ASCO in 2022, and we expect that to be published within the next year. And then finally, S1800D, our non-match study that was unfortunately closed at a futility analysis. We are getting close to having the number of events that we think we can do our primary analysis, and we expect to submit that for ASCO 2024. We have a number of manuscripts in progress that are from the translational medicine group or ancillary projects associated with lung map. We're going to be looking at the ctDNA in the S1800A study. That was the ramucirumab pembrolizumab study that was presented and published recently and is the genesis for Pragmatica Lung. We have a manuscript looking at what drives accrual in lung map, and I'll tell you what the answer is. The more studies we have, the better we do, but we hope to publish that soon. And then Dr. Cosino, who's the TM lead, he has a paper looking at the Foundation Medicine NGS data and has some really interesting results on mutually exclusive and then co-occurring alterations that should be submitted soon and hopefully will be coming out soon. And then we also have a paper looking at ctDNA in cell-free DNA and tissue comparison, and that should help us move towards being able to use ctDNA as an eligibility biomarker to enroll patients into our studies. And then we have another paper looking at immunotherapy biomarkers. So we have a whole host of work that's happening. To go back briefly to the representativeness paper, which was, as mentioned, was published in the Journal of Clinical Oncology, Precision Oncology journal just in the last year. Dr. Vaidya led this, and excitingly, what we saw was that we did better in terms of enrolling patients of older age. Gender was a little underrepresented, but that's because the first set of studies were all in squamous cell histology. But mostly importantly, we did better in terms of enrolling patients of racial or ethnic minorities relative to what you see a lot of industry studies and just in relative to even other SWOG studies, leading closer to what you see in SEER. And so this gives you just a little bit look of the distribution. You can see the percentages of patients in blue that were enrolled into lung map, in the red that were just in SWOG non-small cell lung cancer trials, and then the sort of teal turquoise color is patients who in the U.S. population based on SEER. So you can see the relative comparison of age, and then female versus male, and then on the top, and that we looked at lung map screening protocol versus the lung map sub-studies. And then on the bottom, you can see Asian versus Pacific Islander, Alaskan Native, Black minority, different groups. And so I think that this is a major success story for lung map.
All right. Thank you for that update. We're going to move on. Again, a tight agenda here. Dr. Rakam, can you hear me?
I can.
Good. Now we have an accrual update.
I
Good
think
morning.
there's just one slide showing the accrual.
Oh.
One slide, if you. If someone could man it, there we go. And so this looks at the accrual from earlier in October. And I think that last year we talked really extensively about accrual and how to increase the accrual for LungMap. We have seen lower numbers than historic, in part because we've had fewer sub-studies open. And we always struggle when we don't have an unmatched sub-study. So when we lost S1800D and we are awaiting our next study, as Dr. Wakar will give us some information. But so we're in this place where we have some unmatched, we have some matched sub-studies and are accruing. So the S1900E for KRAS G12C and different subtypes is nearing its enrollment. And we'll hear more about that. And then the S1900G for EGFR resistant tumors with MET amplification is starting to ramp up and just recently opened. So we're excited about those. We're thinking about new studies as as with S1900G, we're looking at EGFR patients, which we had never looked at in LungMap before, and we'll have some MET studies coming along. And hopefully we will get our unmatched sub-study open and be able to increase this. And as Haas alluded to, and we'll hear a little bit more about later today from David Cozano, we are working to increase the platforms and utilize prior testing for some patients so that not all patients who may have already undergone extensive NGS testing will have to have that repeated as per LungMap. So hopefully we'll do some things to improve the accrual. We understand that trials need to be relevant and there needs to be a reasonable ability to enroll onto subsequent trials if you're committed to LungMap. And so we are definitely working on getting more studies and interesting studies into this platform to help increase enrollment.
Thank you for that update. Appreciate it. We're going to come back to some of these issues. Next is the accrual enhancement committee update. And for that, go to Mr. Frank DeSanto, obviously a very important component of what we want to accomplish.
Thank you for the chance to present. The Accrual Enhancement Committee, a large priority of ours over the past year has been re-engaging with the patient advocate community. And so early last year, we conducted an advocacy webinar to bring in advocates, update them on what was going on with LungMap. But we conducted another one in August, and about 100 people tuned into it. It included presentations on sub-studies from LungMap leaders and sub-study chairs. Dr. Jay Nyack, who is an oncologist with ANMED Health Cancer Center in South Carolina, presented on making LungMap work at a community site. And we had a great panel discussion with 3 patient advocate leaders. And some of the things we got out of that were really valuable advice on how to present LungMap to make it more attractive to patient advocates and patients generally. One of the things that actually Dr. Upal Basu Roy talked about was the value proposition of LungMap. And he worded it as LungMap is something for everyone based on the profile of your tumor, which I think is a message that we want to highlight going forward. It talked extensively about what patients need when considering trials, talked about what questions patients have, the need to have some sort of what they called an executive summary that patients could get and take home before they get the informed consent document. And actually that what they described aligns very well with the patient-friendly summaries that we create for all of our sub-studies. And they actually agreed that that was the case. And we actually get significant input from patient advocates on those as we're producing them. The recording of the webinar is online. It's linked from SWOG.org. I think it's linked from ctsu.org as well. It's had about 400 views since. Here's just a shot of the advocate panel discussion. One of the things the advocates also talked about was the importance of the diversity of the research team itself for diversifying trial participants. People are more trustful and more at ease when they're talking to someone who looks like them. Going to highlight the diversity of the LungMap team as well, and the diversity of the enrollment thus far. Dr. Vage's paper, obviously Dr. Redmond has already talked about that, and Dr. Borge referred to the editorial that accompanied that paper, which I'd really encourage you to take a look at. That's in JCO Precision Oncology. It is open access, I believe, and so anyone can get to it. There are some printed copies of it floating around on the registration table with our plain language summaries for sub-studies. Just jumping back again to that executive summary, the desire for an executive summary. We've got these available for all sub-studies. We produce them when sub-studies open. I encourage you to pick up copies at the table, out in the registration table out front. S1900K, we've already got a CIRB approved summary for that, even though it's not quite activated yet. So it's your chance to get copies of that. There is actually a summary in slightly different format for the LungMap screening trial overall as well. I think a lot of people aren't aware of that, partly because it's a few years old. It's in a different format on ctsu.org. It's way down the page. So we're actually going to do a new version in the format of the summary we use for all of our sub-studies. So it'll be more easily recognized. And of course, our patient advocates have endorsed that set of questions in that format. We're also creating Spanish translations of all of our summaries. A bunch of them are actually with CIRB right now for review. Our original goal was to do a webinar like this quarterly, but it takes a lot of effort. It's difficult getting leadership, getting all the necessary people together. So we're looking into trying to do this more modularly going forward, breaking up the recording we have, recording some new modules to attach to it as sub-studies launch, as new topics become important, so that we can do that maybe on a quarterly basis instead. We're also trying to bring, we work with advocates in advocacy organizations that are defined by a particular mutation, for example. When we're working on patient material for sub-studies in that particular area. And one of the things this panel of advocates talked about was the benefit of trying to get those targeted advocates in the process earlier, so that, for example, we had an advocate from EGFR resistors, we had an advocate from crass kickers on the panel, and getting them in at the earliest stages of protocol development when sub-studies are being planned would be a great thing. We've proposed that to the policy committee, and I believe that's going forward. Next steps, starting to plan for outreach when Dr. Cousino's work comes to fruition, and we've got an amendment passed that broadens access to testing assays, which I think is really kind of an opportunity for us to relaunch the trial in terms of publicity and outreach. I think I've reached my 5 minutes. Resources are available. Make sure you're aware of them, pick them up. They're on ctsu.org and swog.org. And I think I've hit 5 minutes, I'm going to stop there. Thank you.
Thank you. So our next talk is going to be on S1900E. I have here Dr. Reckham, right? Okay. Dr. Reckham, are you on for 1900E? I can advance the slides when you're ready.
That would be great. So yes, 1900E is our phase 2 study of sotorasib in previously treated patients with KRAS G12C, looking at co-mutations. Next slide. So here is the schema. And again, there's no randomization. All patients receive sotorasib, and we're looking specifically at co-mutations with TP53, STK11, and then there's a bucket for all others. We're hoping for 40 for the TP53 and others, and 25 for STK11. Next slide. So we're at 101 of 116 participants, 44 for TP53, 22 STK11, and 35 for others. So we are hoping in the next number of months, we will get these accruals and be able to follow for maturity and have additional information. They are also looking at the ctDNA and putting in the TM amendment so that can be evaluated quickly once the enrollment is complete. We do have the ethnicity on these patients. We have 87% are white, 10% are Black, and the most are non-Hispanic. So that's the update. I think that might be the last slide. Oh, one more. Available slots, several for TP53, multiple STK11 and others, but again, nearing completion. And the chairs, Dr. Pata at Temple and Fox Chase, and Dr. David Gerber at UT Southwestern. The questions, if you have questions, on the right-hand side are the links. Thank you.
Okay, thank you. Everybody's a little bit quiet. Want to make sure nobody wants to bring anything up right now. Again, promise we're going to get to your questions at the end, but we just want to finish the presentation. Dr. Goldberg is our next presenter. Bless you.
Bless you. Hi, everyone. Good morning. So I'm going to be talking about S1900G. You heard a little bit about it a few minutes ago during the accrual updates. We just opened a few months ago and really excited about this study finally being available for patients. So just a brief moment of background, since it's a relatively new study, just for those of you who aren't familiar with it yet. This is a study for patients with EGFR mutant lung cancer who are progressing on osimertinib. And we know, maybe you can see in the pie chart there, the frequency of MET amplification as a mechanism of resistance to osimertinib is actually quite common. It differs in different studies, but it's somewhere around 20%. So about 20% of patients who progress on osimertinib will have MET amplification. And we know from prior data that combining an EGFR with a MET inhibitor can have activity. And then this study is asking whether the addition of a VEGF pathway inhibitor, ramucirumab, can also add to the efficacy. Here's the schema of the trial. Again, it's for patients with advanced EGFR mutant lung cancer who have MET amplification as a mechanism of resistance. They have to have had one prior EGFR TKI, and that has to be osimertinib as the most recent prior treatment, but any number of prior lines of therapy is allowed, again, as long as osimertinib is part of or solely the prior treatment. So they could have had chemo, they could have had immune therapy, but it's not required. For now, there is an exclusion criteria for patients who've had prior MET inhibitors or VEGF pathway inhibitors, such as bevacizumab or ramucirumab. That's something that could change in the future, but for now, that's in the protocol. And patients with untreated asymptomatic brain metastases are allowed. And the randomization is to the triplet of capmatinib, which is a MET inhibitor, osimertinib, an EGFR inhibitor, and ramucirumab, the VEGF-R2 pathway inhibitor, versus the EGFR and MET inhibitor alone, with PFS as a primary endpoint. I'm just going to skip ahead and not go through the eligibility, but I did just want to talk for a second about biomarkers, because this is something that comes up. Well, it's already come up a lot in the last couple of months since we've opened the study. So for these patients, EGFR mutations are what's detected at baseline before they even start on osimertinib. Otherwise, they wouldn't have received osimertinib in the first place. But MET amplification is only detected at the time of progression on EGFR inhibitors. So that's the more recent biopsy that's required, or ctDNA, which we'll talk about. So there's a few ways that we could detect MET amplification, and I'll try to go through it briefly here. One is through kind of what I call a standard lung map procedure. So if a patient is progressing on osimertinib and hasn't yet had testing done to look for mechanisms of resistance, you can get a biopsy and submit the tissue to lung map. If they've had a biopsy and Foundation 1 CDX tissue-based testing is done and shows MET amplification, you can just submit those results and you don't have to submit additional tissue. On the other side of this line is if you've done local testing. So local testing could be a tissue-based assay, and that's really any assay that you do at your site, as long as it's CLIA certified, or it could be blood-based. So a lot of our patients these days are getting blood-based assays at the time of progression to osimertinib. And you know, at our site, we do a lot of Guardant ctDNA testing or Foundation ctDNA testing. If that's what you do at your site, fantastic. If it shows MET amplification, they can be enrolled and screened for this study, and you just submit that data. And if you have tissue, great, you could submit it. If you don't, that's also okay. So all you necessarily need is ctDNA if that's what you do as part of your practice. Happy to answer more questions about this, as it often is a point of confusion. So just for very brief updates, I have 2 patients accrued here. These slides are a couple weeks old. We've actually had a few more patients registered even in the last few weeks. We've had 2 at our site, which we're really excited about. We did create this FAQ document to try to address some of the questions we were already seeing. I have a little snapshot of it here, but it's also available for you if you wanted to look at it in more detail. It answers questions about screening for MET amplification, prior lines of therapy, the types of patients who are eligible, and those sorts of things. But of course, always reach out if you have questions. I'm always happy to help. And there's our contact information. Thank you so much.
Thank you. That's great. So remember 1900 G for Dr. Goldberg.
Exactly.
G for
That worked out really well.
Dr. Herbst.
I think you and I talked about it.
G for Gandera? No, the question I have is the Mariposa trial that's going to be presented at ESMO next week. None of us know the results. There's Mariposa 1 and Mariposa 2. Any thoughts? We'll have to just keep an eye on how that might change the space. I don't know that they've checking for MET in those trials, but it might put something out there that could hurt our randomization.
Yeah, it's a good thought. I think just in general, there's a lot changing, or at least a lot of new data coming out in EGFR, both first line and at resistance. And so the landscape could change. I think this question, will still be really relevant because it's looking at a biomarker at the time of progression. So even if someone got a different first line therapy, it still could be relevant. But I agree, it's something we'll have to keep an eye on.
Yeah, I think it's looking at a biomarker, which I don't think the others have done. It's also think about quality of life. Look what Frank was talking about, and patients having to go to an IV infusion every 2 weeks versus an oral agent. So I think there's a lot of positives, but I think we need to get our messaging out for that.
Yeah.
But it's great that this is moving forward.
I agree. I think we have to be prepared for that.
Yeah.
Really good.
That's a great point.
So great presentation. I just wanted to clarify that the G is for gray.
This works
But
out really
I'm sorry, I couldn't resist.
So many.
That was actually that's not why I got up here. So I wanted to ask, I love that you have the broad eligibility criteria, right? Any local tests that has been approved within your protocol, you can use that cut point for the MET to enroll the patient. I think that's the right way to do it. My question is, in the background, are you also going to have certain thresholds that you're assessing to do some correlations, if you can talk to them about?
Yeah, so we talked a lot about this when we were developing the study. Should we actually use a cutoff initially to enroll patients and for eligibility? And we decided against it for a few reasons. One, we don't really know the right cut point. There's been studies looking at it already, and I think there's a bit of conflicting data. And then the other thing is, and this is probably the more practical reason, is the ctDNA assays don't even give you a level. They just say amplified or not. And so we really don't know how amplified those tumors are. So for eligibility, any level on tissue or just being called amplified on ctDNA is eligible. But yes, we're going to go back and look, and that's going to be a really important part, because I think the MET inhibitors I hope will have some role in the future in these patients. None of them are standard yet, but the cutoff, I think, is going to really
help us identify the patients most likely to benefit, right? The higher the amplification, most likely the more likely there's going to be benefit. And so, yes, we're going to be looking at it is the answer. Thank you.
Great, thank you.
Thank you.
Our next presenter is also going to be virtual for S1900K, Dr. Paul Pack. Paul, can you hear us? He did have a very tight schedule. I can't hear you. Hmm?
She's asking.
Is Dr. Lee on? I didn't see her name there. I checked earlier. Yeah. Unfortunately, we don't have the PI or the co-PI. We knew they were under pretty tight schedules. Dr. Pak, are you there? I don't see his name on the list. Are we early for him? Could be with the patient, yeah. I can come back to this and I can definitely present the update, but let's give Dr. Pak, another minute or two to see if he can join. In the meantime, why don't we go to S1900J and invite Dr. Christian Rufo, who was in the audience. He just left out too. All right. I think I'm going to go ahead and talk about the 1900K then. So this, as the title talks, is a phase 2 study, tepotinib, with or without ramucirumab in patients with MET exon 14 skipping mutation, stage 4 or recurrent non-small cell lung cancer. The study chairs and champions are shown on the slide here. So MET alteration in non-small cell lung cancer is something that I think we've discussed at several meetings. We understand that the exon 14 skipping mutation is a particular alteration that's detected in NGS. It is a rare subpopulation for sure, but the majority of the platforms out there are able to detect this particular alteration on an NGS broad spectrum panel. This is an actionable mutation. There are 2 different drugs that are already available for patients with this diagnosis with really good clinical activity. One is tepotinib with an overall response rate of around 46%. This is on all lines of therapy. And the next one is capmatinib, also with a response rate around 41% in the second line, 68% in the first line setting. The PFS is anywhere from 9 to 12 months with these drugs. And the waterfall plots just indicate the robust clinical activity that's seen with both of these drugs. Doctors Pack and Lee were also the authors of the New England Journal of Medicine article with tepotinib, so they have a lot of experience with this drug. This is the NCCN guidelines that basically recommends treatment with a targeted agent for patients with a MET exon 14 skipping mutation, just to highlight that this is considered to be the standard of care. The efficacy of several different targeted therapies are shown, along with the potential for PFS. And again, the reason we're talking about all of that is that as good as tepotinib is in this patient population, as you can see by sort of this diagram, we're not approaching the kind of clinical efficacy we get with agents like alectinib or osimertinib. Sort of these have become our go-to drugs in terms of clinical efficacy, durability, toxicity. So there's a lot of room for improvement for patients with MET exon 14 skipping mutation when it comes to the use of targeted therapies. Now, don't get me wrong, we're happy with this. It's better than cytotoxic chemotherapy. But again, our goal is to find better treatment options. So the goal is to move tepotinib, if possible, to sort of in the far right-hand corner of the graph. We understand that these drugs come with toxicities, unfortunately. We've talked a lot, again, at various meetings and also in the publications with these agents regarding the potential side effects. And one of the bigger one is the peripheral edema. As you can see in the treatment naive patient population, up to 58% of patients developed peripheral edema. And, from a clinical experience, this is a difficult side effect to manage. There's no doubt about it. It is something that many of our patients have to deal with when they go on these targeted drugs. And the median time to onset of some of these toxicities are very similar among different drugs. But, as you can see on the left-hand side, time to the first onset of peripheral edema is very short. On the top, you see weeks, around 7.5 to 8 weeks. So this series of toxicities gives us an idea that these are going to have an impact on patients' quality of life because they're not exactly going away. And we do have to come up with ways to try to mitigate that. And again, these are highlighted by the durability of some of these adverse events that the patients have seen. So the rationale here is that there's potentially a synergistic anti-tumor activity between a drug that targets the MET exon 14 pathway with an anti-VEGF. So the dual inhibition, based on some of the animal studies that you see on the slide, also could be better than the single agent MET targeted therapy. The signaling through the VEGF pathways also increased after MET TKI use, which leads to some of the potentially structural changes in the endothelium that we think could actually exacerbate the edema that's seen with these drugs. So this is a lymphoangiogenesis process that we've talked about. So in fact, we think with the VEGF inhibitor on top of the drug, not only we should have better clinical activity, we're hoping that we'll see a little bit of a less of an edema. So the hypotheses are listed. The primary mechanism of resistance to MET inhibition and peripheral edema is as a result of upregulation of the VEGF and VEGFR2 pathway, which is induced by MET inhibition, and then coupling of ramucirumab to tepotinib could increase anti-tumor efficacy and also improve tolerability. This is the schema, standard eligibility criteria for patients. Treatment is going to be tepotinib, the standard oral dose plus ramucirumab at its standard weekly, 3-weekly dosing. And the control arm here is going to be tepotinib. The primary endpoint is going to be overall response rate. CNS MET are allowed if they're not causing symptoms and they're less than 1 centimeter. Obviously, we do not want to have any contraindications or use of anti-angiogenic agent, and patients have to be MET inhibitor naive, which again, I think kind of makes sense for the kind of study that we're doing. This is a statistical analysis plan and consideration for the study. We're estimating the response rate to tepotinib alone is about 45%, with 25 patients per arm, with 90% power to detect a response rate of 34% at the 10% level. So difference in response rate, I'm sorry, 34% difference in the response rate. These are the study chairs. And again, most of our protocols have a very handy front page where you can find everybody who's involved with the protocol to contact them for any additional information. And I think with that, I'm going to stop if there are no immediate questions. And I'm wondering if Dr. Rufo has come back. If not, I have his number.
Can I ask a question?
Sure.
So, Haas, I realize you're not the PI of the tepotinib study, and maybe I missed it, but there's several recent publications about mechanisms of resistance to MET inhibitors with exon 14 skipping. And just like everything else, there's secondary MET mutations and their bypass mechanisms. So this is a great opportunity, even if we do it with ctDNA, to look for the mechanisms of resistance. And maybe rebiopsies. I didn't see that in here. Is it?
Yeah, so
This study has ctDNA assessments at baseline. I think, after cycle 3, day 1, and then at progression.
Yeah, definitely. All right, we found Dr. Rufo, as you see, standing here. I'm going to ask him to run through S1900J.
Okay, thank you for the opportunity to discuss this trial. We are working with Dr. Shirish Gadgel, and you see here a group of people that is supporting us, Dr. Gray, Dr. Gandara, and Dr. Redman. So this is a phase 2 trial with amivantamab in patients with the stage 4 recurrent non-small cell lung cancer, previously treated with high MET amplification. So as we know, MET amplification, MET is a very important pathway and very complex pathway that we can have there. I know that are a reference on the Greek mythology. For those that doesn't know, the Hydra was a mythological animal that when you are cutting one of the head, appear others, and that's what's happened with MET. So we have there opportunities to tackle the MET mutations, the MET amplifications as a mechanism of resistance, but also MET amplification as a primary driver. So this is the meaning of this trial. The prevalence of the target as a primary driver is in 3 to 4% of all the non-small cell lung cancer. And recently, there are also some data on squamous cell lung cancer as well. So this is a big opportunity for this unmet medical need. So the amplification in say can be targeted by multi-kinase or MET-specific inhibitors. And we saw in the historical cohort, for example, with crizotinib, that the data were around 30% of overall response rate, progression-free survival 4 months, and overall survival around 9 months circa. So amivantamab have an important activity in MET and EGFR, and data obviously working in the MET exon 14 skipping mutation, but also in other aberrations of MET. And that's, you see there, the waterfall plot of the combination with lazertinib, show an activity in patients that have even MET amplification as a mechanism of resistance. So there is a rationale behind that we can see that this drug is working in MET amplification as a primary driver. The study will be enrolling 80 patients, so 44 in total for each arm in these eligible patients with the non-squamous and squamous cohorts. In the beginning, we was discussing also to include liquid biopsy as a possibility of a stratification or a selection of patients for accruals. But the problem of MET amplification in the liquid biopsy is the quantification. Foundation found a way to select the patients with more than 6 copy numbers. So that's an important opportunity to identify actually the high amplified patients. So we will collect the blood sample, but the main strategy to accrue patients will be the tissue samples, and we will perform actually through foundation. The primary objective in both cohorts is the overall response rate confirmed by the RECIST, and secondary endpoints will be including progression-free survival, overall survival, duration of response, and the toxicity of this compound that we know that have some infusion reactions that we want to also see what happened in these specific cohorts. The liquid biopsy part and the tissue biomarker part will be also part of the translational component, and that obviously will depends on the approvals of this part, but it's a good opportunity to not only see the detection in liquid biopsy, but also the opportunity to monitoring the response of these patients with the ctDNA and the dynamics of the BAF. So inclusion criteria, and I'm sorry that the last slide was, no, you completed, thank you. The inclusion criteria are including, as I say, patients with MET amplification by FMI. We can include also, and that's a good thing, that we can include patients with MET, with CNS metastasis, asymptomatic. And also, obviously, patients need to have exclusion of all the other biomarkers in order that we really exclude patients that have this MET as a mechanism of resistance. The prior and current therapy, we include patients that at risk have a progression of 1 line of treatment. And this is the trial status at the moment. So we have the approval in September. CRB initial review, and I saw this morning that we received something extra, some comments, and it's the approval, final approval seems to be. Just comments. Very minimal comment, so easy to respond. Thank you. Because I saw the email this morning and the activation is target for December 1st. So we hope that the scenario, because actually it was a very short time, is my first trial in working with LungMap, but was an excellent experience, and the team with Jennifer Biller and all the team that the professionals that are working behind is really interesting, and was a great opportunity to see how we can speed this very quickly. Thank you.
Thank you, Dr. Cooper. So I have a question, Dr. Gray.
So just a similar to what I asked Dr. Goldberg here about, again, your cut point for the assay. My understanding is that we spent a lot of time thinking about amplification and looking at the cut points, and your cut point is a little bit different, right? And I think within the umbrella of LungMap, I think it maybe you can tell us a little bit more about how you chose your cut point for MET.
So you can calculate the MET amplification with the ratio of the amplification, or you can do directly with the copy numbers. And we selected the copy numbers by FMI, that is 6 copy number above. So that will be really the corresponding with the high amplifier. And that is also similar in liquid biopsy. You can get the same definition.
The 6 copy number comes from the 6 copy numbers came from a publication by Ross Camich and the Colorado group some years ago, where they looked into MET inhibitors and looked into different cutoffs, and they ended with 6 as the highest.
Yeah, and actually that's a very good comment. We saw presented recently last year in ESMO data on squamous, and they were using another kind of definition. So we don't know actually the real presence in squamous, because you can have also some false positive there using different techniques. So we will see, actually this is a good opportunity also to include the squamous and see how is the presence and the prevalence of MET amplification in this population.
Great. One more question?
It's known that, we talked about the umbrella protocol for LungMap, so highlighting that.
Yeah.
If I understand this correctly, some patients will have received immunotherapy in the first line, and then curious about like the timing in terms of second line capmatinib, in that like we know there may be a risk of like ICI toxicities. I'm wondering about like whether some window there, maybe.
Yeah, we also have so there is a normal, I think, 2 weeks between the 3 weeks between the treatment, the last treatment, but this is an antibody is different to capmatinib, to a TKI. So we will not expect it to have special reactions in terms of pneumonitis or other events. So we are a little more scared about the infusion reactions than other things. So, but it's a good question.
Okay, thank you. Appreciate it. All right. We're going to move on to the next stop, and this is obviously the contact information. So I think Dr. Redkamp is on also. We're going to talk a little bit about what we're going to be doing in terms of the NGS platform expansion. So this work was primarily carried out by Dr. David Cousino. We're going to hear from him next. He is virtually available for the next section, but all the credit for changing and going through this process of allowing other platforms with NGS testing to enter LungMap is as a result of his tremendous amount of work. So a big thank you to Dr. Cousino. Dr. Redkamp, if you want to interrupt and say something, please go ahead and raise your hand and I can give you the podium. So where we are right now is that, as many of you know, we traditionally have been using the Foundation Medicine as our only platform, and even if patients had testing at other institutions or other places, we sort of wanted to redo everything just for uniformity. I think this has a historical background, obviously. When LungMap was started, NGS platforms were not very common. Foundation Medicine was the most extensive NGS testing platform available. If you remember, again, way back when LungMap was started, we were concerned with developing specific targeted therapies in patients with squamous cell histology. Routine molecular testing was not customary for patients with squamous histology. So there were a lot of historical reasons why one platform was chosen. Obviously, things have changed. We realize that many different institutions do a lot of testing on their own. We also realize that there are other platforms where our colleagues send tumors for molecular testing. And we heard from many of you that this was a barrier for patient enrollment, need for repeat biopsy, patient going through waiting for molecular testing to be done. And in order to streamline everything and improve accrual and also to be more sensitive to the needs of our sites, we decided to expand the available platforms. Dr. Cosono and the group met with our colleagues at NCI Match, where, again, for those of you who are participating in that mechanism, it requires extensive testing. So they had gone through this process of basically validating many different platforms and institutions where the experts on the NCI panel felt that the molecular testing was appropriately done. So we met with them, talked to them about their process, and through a series of very long discussions with various groups, we now have a protocol that is the screening protocol that's going to be changed. We're going to be allowing multiple different platforms. We have specific criteria for what we are looking for in terms of the validity of the testing. And we're hoping that with this addition, and when the amendment is finally approved, we will be able to include a lot more of your patients on a study and not have to rely on retesting with the Foundation Medicine platform that we have been using up to now. I don't want to go into the details of all the selection criteria. However, if any of you have questions, at the end, we are more than happy to sort of go through that. And again, I'm hoping that Dr. Cosono can stay for the Q&A part in case a question comes up. But this is the latest sort of change to the screening protocol. The amendment has not been submitted. We're still going through a review process. We've asked our patient advocate on the committee to actually review the protocol as well, just to make sure that everybody agrees the screening is as pragmatic as possible. But we're hoping to submit the amendment very, very soon and have this option available for everyone. Dr. Rakam, any other comments?
No, I think you've covered it, and Dr. Cosono is going to cover it. So I'm going to let you all let Dr. Cosono talk, and then there'll be more time for questions at the end. Thank you.
Great. So with that, I am actually going to invite Dr. Cosono to join us virtually. And David, thanks for joining. I can advance the slides. I know you're going to provide us an update on translational medicine, but perhaps we can also cover the NGS testing that we just talked about.
Absolutely. Real pleasure to be joining you here. I'm just a couple blocks north of the White House right now at a meeting here, so sorry I couldn't be there today. Next slide, please. Next slide. And so I think this is covered very well by Hassa and Karen's introductory comments, but this really is indeed the push for expanding LungMap to provide options for our patients based on how things are done here in 2023, where people are having NGS testing done by a variety of commercial and academic platforms. And it is indeed a barrier to ask patients to have this retesting done for the sake of our Foundation One CDX mechanism. Next slide, please. And so our group took a close look at what makes for a good test for LungMap, and we decided that it's potentially any CLIA certified test that's sufficiently broad. What does sufficiently broad mean? It'll have to include all of the alterations we've been interested in so far in LungMap, such as those genes that are listed here. And it generally comes down to it being a panel of at least 150 genes, that was a cut point at which we can expect to find essentially all of the genes that are listed here. And also the point at which we can start calculating composite scores, such as tumor mutational burden, which is also an important biomarker these days when it comes to immunotherapy. I want to stress right up front that we don't want to limit the number of tests as long as they can meet criteria. And so we have a process in place for doing real-time qualification of any new CLIA certified tests as they come on board, and so that this will be a continually updated process. Next slide, please. But just for now, here are the commercial lab tests that we felt after looking things over currently meet criteria. And I hope this represents, 90% of what people are doing when it comes to commercial tests. So the likelihood is that this will cover the patients that you are currently seeing. But if there are additional tests that seem like they would meet criteria that we don't have on this list, please reach out to me and my group anytime. Next slide, please. You can see here a longer list of academic labs that also have tests, whether targeted panel or NGS, that also appear to meet criteria. And so we're going to start with this list of sites. But if there are sites at which you are based, that are not on this list, I'd love to hear about it right away, and we can see if we can also include you onto the initial drafts. Next slide, please. So far I've been talking about tumor testing, but increasingly patients are interested in circulating tumor ctDNA blood tests to identify potential biomarkers for clinical trials. And it is the case that based on work by Bill Mack and others in LungMap, that there appears to be sufficient positive percent agreement or predictive value to say if you find a tumor EGFR mutation, let's say, in the blood, that this is very likely to also be in the tumor as well, sufficient to the point that this would be a reasonable biomarker for inclusion on an EGFR-specific match substudy. We are closely looking at negative predictive value, meaning the ability of blood tests to exclude the presence of a mutation. And it does appear that there can be a tumor fraction cut point that would allow this on studies. And once we have that fully validated, we may be able to include this as well when it comes to ctDNA. But for now, we are looking at patients who have tumors that are positive for biomarkers for actively growing matched substudies to have the potential of entering based on ctDNA. Next slide, please. So far, these are the two tests that meet our criteria, the one from Foundation Medicine, the F1L CDX test, and Garden 360. But I anticipate that there will be others. And again, please reach out to me if you feel that there is a test you would like included in this effort. Next slide, please. And so what really gave me confidence that this would be, a way we can move forward is, due to, Pragmatica Lung S2302 that Dr. Rickhamp and others are leading, where we are seeing the proof of concept here that sites can fill out these case report forms indicating the presence or absence of these various key oncogenes and tumor suppressor genes in lung cancer. And it seems that accrual is going well and that this is going well as well. Next slide, please. There are, of course, some biomarkers such as MET exon 14 skipping that have rather intricate definitions as the one seen here. I think for our investigations, we'll have to conclude that, we'll have to trust, if a commercial or academic test calls a MET exon 14 mutation, even if it doesn't match 100% the rules here, that we may be able to use that in most cases as still, a biomarker for screening purposes, although there will be the opportunity to investigate the more intricate features about these alterations on the back end. We don't expect sites, however, to have their coordinators understand, these different like intron and intronic splice and acceptor sites, for example. Next slide, please. Now, there can be confirmatory testing of samples as needed. There should be some flexibility based on things like the registrational intent or lack thereof for a particular substudy. But we may, for example, require still tissue if we do need to do some central testing because of substudy specific requirements. And for now, the feeling is that if patients only have ctDNA testing results and there is no biomarker-matched biomarker identified, then we should still provide the opportunity to do tissue NGS to confirm the presence or absence of these biomarkers and also support our biomarker negative studies that way for now. Next slide, please. And so this is a flow chart, but what I really want to really focus on is that I feel that we should continue Foundation Medicine CDX testing for those sites who want to submit tissue. This is, I think, a real service to the community that we've provided since 2014, maybe increasingly patients and their providers don't need this, but I don't feel like this should be taken away because I think there are still, patient populations where this testing may not be as available. We really want to support diversity in our studies. I think that'll be important. But then by now accommodating these tissue and blood NGS testing platforms that are currently in routine use, I think we'll be able to support accrual, and LungMap exists based on the accrual of our patients. Next slide, please. So here I've been talking about NGS testing, but I wanted to say that that's only a piece of the puzzle as we look forward, because NGS testing only reveals the DNA alterations that can impact protein function. Now, cancer biology can be driven by many points along the biological pathway from protein to RNA to DNA to RNA to protein, as you can see in this classical diagram that we see in Intro to Biology. And we have to recognize that the protein expression levels can be affected by processes that we would never understand by NGS testing alone. Next slide, please. And so to that end, we want to understand, the ability to do protein or perhaps RNA profiling, so we can then start having biomarkers for the next crop of studies. For example, antibody-drug conjugate. These ADCs, for example, TDXD and sacituzumab govitecan, these agents often have bespoke antibodies that are matched to the antibodies in the ADCs. And eligibility or certification to various studies may depend on protein expression levels determined by immunohistochemistry. Now, today, people have been doing these one-of IHC testings for these different ADCs, but the future holds promise of many additional possible molecules, and it would be infeasible to be able to do 10 single IHCs based on the tissue that we can obtain. From our site. And so we're looking at ways to do multiplex IHC or mass spec-based analytical methods so that we can screen for the presence of high expression of multiple markers at the same time, so that we can then inform these type of studies going forward. Next slide, please. And another, I think, big area for where we need expression-based biomarkers would be in immuno-oncology. Primary and acquired resistance to these agents may be related not to gene mutations, but rather to expression levels of proteins such as PD-L1, IDO, LAG3, or TIM3, loss of immunodominant neoantigens and the like. And this is a dynamic process that would require us to be able to see what's being expressed in the cells and how that changes over the life of these different therapies, so that we can then start targeting things like acquired resistance and really inform our IO studies accordingly. Next slide, please. And so, these are some of the considerations that I had mentioned. There are additional ways that proteins can be modified, such as with post-translational phosphorylation, ubiquitination, simulation, glycosylation. There are quite a number, and many ways that RNA expression also that can be altered. We are investigating these so that we can see what would be the best platform forward. And then what I see really happening as we approach our companies with this RFI to try to pick a next technology, is this would be like the reiteration of what we did in 2014, where we were able to give the community access to something that wouldn't routinely be available. At that time, it was NGS. Next, it'll be things like a comprehensive protein and RNA profiling. And so that could inform a new battery of studies that need these results in order to find patients that are appropriate for them. Next slide, please. And so for the last part of my presentation, I did want to talk about how we could make use of the LungMap data and biospecimens that we've been collecting. Because not only are we running clinical trials, we're creating a wealth of resources that will inform the next generation of trials based on the biology we learn with every patient we are currently bringing into our programs. So next slide. I did want to mention just in brief, what kinds of samples we have. Of course, we have some of the tumor tissues that have been submitted, especially when sites have submitted tissue blocks, and that's been preferred. We sometimes have slides, but in general, it's the blocks that we can retain for longer durations of time and maintain quality. We have blocks from 1400. And we do have them for a majority of patients on LungMap because most have been brought on based on central testing, although sites that are using commercial results as we started permitting, and not submitting tissue, we would not have these resources. Next slide, please. So I'm happy to say we're about to submit this manuscript that did a comprehensive analysis of our S1400 sequencing data, which had a total of 1672 patients for whom we had data from the old Foundation 1T5 research platform. And we've identified over 36,000 variants among those patients, an average of about 20 or so variants per patient. And this represents the largest NGS data set of advanced squamous cell lung cancers of previously treated patients that exists. And so we're going to be publishing a primary analysis of this with a few interesting findings. But my hope is that people will be able to use the data set as we release it with this manuscript to do additional secondary analyses and also to really understand, the genomic landscape of squamous cell lung cancer and its advanced manifestation. Next slide, please. This is a little bit of a dense slide, but it has to be dense because it shows the very rich set of blood samples that we've collected across our studies, including the older S1400 series and the newer S1800 and 1900 series. You can see that we've universally chosen to collect samples at baseline and at multiple fairly consistent time points for generating EDTA plasma frozen aliquots. And the vast majority of these have not been employed in various studies yet. We're very much keen on soliciting proposals that would make good use of these samples. And we do have a very rich set of samples with a good variety of different molecules tested for study. We don't have as much in the way of cfDNA samples for the older S1400 series, but you can see that as a almost consistent rule, we have pre-treatment and first progression samples across all our currently active S1800 and 1900 series studies, which represent an excellent resource as well. Some of this is being tested so that we can look at things like central, the concordance between, tumor and ctDNA detection of alterations, but there will be sample available for additional analyses as well. Next slide, please. And so I want to give a couple illustrations about how we can make use of every bit of resources that come into the LungMap program. And just by way of example, one of the requirements when submitting tissue is to send either an H&E slide or at least one extra slide so that Foundation Medicine can do that H&E testing. I'm now working together with Brian Hennick, who's going to take the lead on a project to take those H&E slides and see if we could learn about what kind of tumor alterations patients have using technology from an industry collaborator, Imaging, who's going to be taking a look at those slides using AI and machine learning to try to identify things like EGFR, alterations and other tumor oncogenes and suppressors, just based on the H&E slide alone. Long ago, when people looked at these H&E slides of lung cancer, the first step was saying, this is small cell or non-small cell. Later on, we broke down non-small cell into things like squamous and non-squamous. But it turns out with enough examination of fine detail, one can actually see, oh, this is a tumor that's being driven by EGFR, MET, RET, etc. And so this may be yet another way we will be able to offer screening to direct patients to the right therapies for their cancers. And so I think a wonderful example of TM research that we currently are embarking upon. But we also have other things like these residual DNA samples. And so we really want to crowdsource innovation here. So I try to make this as simple as possible with the one-page letter of intent that I would like to see widely distributed. Please use this to just kind of indicate what sorts of samples you might be interested in so we can see if it's the sort of thing we have and what kind of analysis you'd like to do. Really not meant to be onerous at all, just the first pass to see whether LungMap might be right for a TM idea. And this way we can then solicit these proposals, vet them in our committee, and then really help with the development of a proposal that we could then present to CTEP for approval. Next slide, please. This is just acknowledging that there are many stakeholders involved in LungMap, but rather than seeing this as being multiple redundant steps review, I'd instead say that there are key insights that our operations team, our statisticians, and our scientific leadership can provide to really make sure that the best science is being studied in your proposals, and that we can ensure the highest likelihood that it'll get full approval when reviewed by CTAP. Next slide, please. I just wanted to thank the group, and this is my email. Please indeed email me anytime. I'd like to hear about any ideas you have as early or late in the process if you care, but definitely want to help in any way I can. Thank you.
Thank you, David. Really appreciate it. In the interest of time, I'm going to ask, okay, one
Can I ask
question there.
Quick question.
Yeah.
David, it's David Gandera here. Before you get 1,000 requests for these samples, it's my understanding that CTAP will not approve discovery, exploratory, that it needs to have preliminary data and be hypothesis testing.
That's been my experience as well. So we've had a number of proposals that unfortunately we were not able to move forward with for that very reason. But I would say the bar is different depending on what you're asking for. If it's going to be for some of our precious limited tumor specimens that are banked, yes, the bar will be high, and that is traditionally what CTAP is looking for. But for example, the reuse of digital H&E images is a project I just mentioned. That's the sort of thing that I feel we can make the case that even with a bit more discovery work, it's not like you're exhausting a resource, and therefore maybe entertained.
Right. So next is Dr. Walker, who's going to talk about the Drug Selection Committee. Obviously, this is a really important committee. Without the efforts in this committee, we would not have any sub-studies. So it's a lot of work that goes into this. Dr. Walker, are you on? Yes. Yep, I can hear you. So I can advance the slides when you're ready.
Appreciate it. Thank you very much. Thanks for the opportunity to speak to the group today. As Haas just alluded to, I'm the chair of the Drug Selection Committee. I'm a medical oncologist at Washington University School of Medicine in St. Louis. Next slide. Here are my disclosures. Next slide. So this is just an overview of our Drug Selection Committee, and there are several sources of ideas for the sub-studies. Some of the sub-studies, as you know, are match studies, and so are biomarker driven, and some of them are non-match. These either can come from investigators based on preliminary work that they have performed, or this can come from pharma approaching us. Most of the studies that we're looking at for LungMap, they have to have some data at least which is ready to enter into phase 2 level before they can actually be used in the drug selection committee. So there are times where studies are kind of halted, or we're waiting for more data to emerge, and before we can actually proceed ahead. So they can be pharmaceutical company initiated in terms of ideas. They can be investigator initiated as well. And then the composition of the drug selection committee is shown. There are key investigators, clinical researchers, biomarker researchers, experts from academia, NCI, FDA, and non-conflicted industry-based drug developers as well. Next slide. So this shows basically the process. So first of all, I mentioned the different sources of the sub-study ideas. Initially, there are internal drug selection committee calls set up where we look at what is the data so far, and what is the study that could be proposed. How can we fine tune that idea a little bit further so that by the time that there's a full drug selection committee presentation, it's pretty fleshed out. There is a process for submission for drug selection committee. There is an application form that has to be submitted that outlines the agent, the data so far, toxicity, biomarker understanding, clinical data, and then that's submitted prior to the presentation. Once the presentation occurs, actually for the actual presentation, pharma is present as well as the investigator. There's a Q&A session, and then they leave the meeting and there's a closed committee discussion. Following this, there's formal voting by the drug selection committee members. There are specific criteria that are each scaled from 1 to 9, which are looking at the target appropriateness in non-small cell, the drug and biomarker understanding, preclinical data, PK/PD data, toxicity, and then the clinical data. If this study is approved by the drug selection committee, then the acceptance letter is sent. And if there is some further tweaking to be done, that information is sent to the investigator as well as to pharma, and then they can fine tune that further and then represent. Once the study is actually approved, then there's a series of calls that get set up. And these occurred at regular intervals, often every 2 weeks, basically to develop the concept and the protocol further, looking at the regulatory aspects, FDA requirements, and obviously also be able to confirm that there is a drug available for the study as well, so that there's not a drug supply issue. Next slide, please. So this shows the current schema for LungMap, and you've heard today about the S1900E, which is open with sotorasib, and from the other TKI-treated study, S1900G, looking at MET amplification. That study was also presented. There are several studies that are listed in blue over here, both in the match and the non-match space. You heard about S1900J and S1900K already, but I will touch briefly upon S1900I and S1800E in the next few slides. So Haas, we can skip this one since the study was already presented, and S1900K as well. So S1900L, so this is a study that was actually accepted by the drug selection committee. It was approved by them, but it has to come back because of some retweaking. So this is a study looking at CEACAM5 ADC, tusimittamab ravtansine, and CEACAM5 positive previously treated stage 4 recurrent non-small cell lung cancer. And this was specifically for patients who had expression noted as greater than 2+ intensity and greater than 1% TPS, so moderate or high. The frequency of moderate is about 30% in adenocarcinoma, and for high, it's about 24% in adenocarcinoma. In squamous, high expression is pretty rare, but moderate expression is seen in about half of the patients. And the patients were going to be randomized either 1:1 to receive pembrolizumab with tusimittamab ravtansine and ramucirumab versus the arm B was kind of looking at the effect of pembrolizumab and ramucirumab, looking at S1800A regimen and comparing that here. The idea was to isolate the effect of the tusimittamab ravtansine. And then there was a single arm exploratory cohort as well in this study, which was looking at patients who actually had driver mutations but had exhausted all lines of standard therapy. Prior immunotherapy was required for this one. However, we have run subsequently into drug supply issues and issues with budget, due to which a redesign is being proposed. Can you go to the next slide, please, Haas? Where the exploratory cohort is going away, and then the arm B is being requested to change the docetaxel ramucirumab as that is approved, and maintaining arm A as is. This has gone back to drug selection committee, and there will be a formal voting to see if this moves ahead or not. Next slide. This is S1800E. It's our non-match study. This was drug selection committee approved, and then there was a redesign, switching this from a phase 2 to a phase 3 study. And this has been submitted to CTEP on September 21st, and we are waiting for their comments back. This, as I said, is a non-match study looking at patients who were treated with chemotherapy and immunotherapy. It's the same study population as S1800A. So if they have been on immunotherapy, it has to be for 84 days or longer. They get randomized 1:1 to receive either docetaxel ramucirumab, which is the standard of care, or the triplet regimen continuing immunotherapy with the chemotherapy docetaxel ramucirumab. And there's a 10-participant safety run-in for this arm B. The sample size is 378 patients in order to get 340 eligible patients. And the primary objective of this is to look at overall survival. Next slide. So there are several other studies that we have which are going through drug selection committee that are a little earlier in development. In the interest of time, we'll skip over those, but just to summarize, we have immunotherapy studies, studies looking at antibody drug conjugates, studies looking at targeted therapies as well. And we would urge all investigators who are interested and proposing a study to reach out to us. This, this study design can come from like a junior investigator. We can link you with a senior lung map investigator as well, and we can fine tune the study and then have a drug selection committee presentation.
Thank you very much, Dr. Bogart. There's a very good overview of drug selection committee. So I have a request online to go back to one of the slides that Dr. Costono showed with the names of the platforms that are allowed. So I'm going to go back to that, but I'm hoping that we can provide that information to everybody who needs it. And also, once the protocol is amended, it's going to be in there. But since this was a question, I just want to go to. So these are the commercial labs where the testing is done. I think this is the slide that you were asking for, Ms. Karen Wishnow. But the forum is open now. I mean, we have a few more minutes to talk about any questions, any issues, any problems that you might have noticed with LungMap. So please come to the microphones. Don't be shy. It's a friendly group. Most of us anyway. And we want to hear from you.
I'm not sure that this shows the latest version.
Of the companies?
Yes.
But this is what we got from David. So David, do you have a comment about that?
This is our initial list that we put in the current protocol draft, but these are just the commercial, and then these are the academic labs. If you total everything, it is over 30 tests.
David, I'll send you a note.
Okay.
That'd be great.
So we're going to update that. More inclusive, the better. Okay, good. I don't want to keep everybody here, but again, I'm going to be here for another couple of hours. Dr. Redman, Dr. Gray, Dr. Gandera, we're all around for the duration of the meeting. So come and find us if you have any questions. I'd really appreciate all of you being here. Thank you for your continued support, and we look forward to the continued success of this program. Thanks.